This is you. You ready? So I'm going to pass you to Gordon now. I should Lloyd's
What's that?
Oh, Lloyd's again.
Okay, that's good, that's good. So, yeah, good afternoon. Gordon King from Taylor Shellfish. Helen wrote me that we were doing this to give a bit of industry perspective. So, shellfish agriculture on the west coast, both in BC, Washington State, Oregon and California, has actually been happening for well over 100 years. It's provided a lot of rural jobs, and to be honest, it was probably almost as big an industry 100 years ago as it is today. If you go around the world, as Helen mentioned, mussels in particular, you're in a lot of different countries. Large production in China, Spain, Chile, Philippines, New Zealand, Ireland, Prince Edward, France. So, you know, mussels, although they really are, as are oysters, somewhat of a weed, like weeds, they don't always grow as productively as you'd like them to. And if we can make mussels more productive and more successful, then we can provide better income in these often remote and rural communities, and provide both shellfish production for domestic consumption and for export. The company that I work for, Taylor Shellfish, it has about over 300 employees in Washington State, about 80 employees in British Columbia, and about 30 or 40 employees in Fiji. And we grow a variety of different shellfish, but we started growing shellfish, started buying into the industry in British Columbia about 6 or 7 years ago, and then a couple of years after that, Helen approached us about partnering with them to try and work out what was happening with the mortalities that we see in the species of mussels that we grow here. And in British Columbia, we're trying to grow Middle's Gala provincialis and Middle's aegilis, two different species. As part of that, Helen asked us to provide her with pure groups of both species. It turned out that the Middle's Gala provincialis that we provided her was pure, and the Middle's aegilis that we provided her was kind of a classic hybrid of 25% pure galas, 25% pure aegilis, and 50% crosses. And that in itself was very helpful to us because it helped us understand why at times we couldn't tell them apart, and they exhibited very similar characteristics. So Helen has over the last few years developed these microarrays which could hopefully tell us, or should be able to tell us, what stresses our animals are under, which is causing them to die. If we can get them to survive, then with the many hundreds of miles of coastline in British Columbia are suitable for growing suspension culture shellfish, hopefully we can see a larger industry grow here. Some of the native bands here are interested in suspension culture of different shellfish, particularly mussels. So it could be beneficial for a lot of people and bring a lot of income in this area. Then another issue that's happening right now is ocean acidification. In our hatcheries, at times, the last few years, we've had high mortalities with goula particularly, and oyster larvae, and that deeply affects the production along down the road, because without the juveniles we can't get the adults. In the future, possibly, we would be looking to use similar techniques, microarrays, to try and determine what stresses our larvae are experiencing in the hatchery. You can do histology on larvae, but it's a little tricky, and using genetic systems would be very helpful. So I'll leave it at that.
I think you've got a question or slide.
Okay, yeah, we can do that. Right. So yeah, and so Helen just threw these in for me, talking about this ocean acidification, and yeah, and as I mentioned, that is a serious issue to us. That's cost us a lot of money over the last few years. We're only part of the industry, of course. There's a lot of other people. There's a lot of farms in BC, and a lot of them are not very economical, and a lot of that's to do with survival, both mortality and productivity. So having another thing which I meant to bring up was that compared to other forms of agriculture, mussels are, oops, went the wrong way. Mussels are really at an early stage of husbandry. With cattle, you see very divergent breeds of cattle. With shellfish, basically, we have different species, but not very much differentiation within the species. There's a little bit, but by breeding and then testing with microarrays, the response to particular environments that the animals we're trying to grow their animals in will then very quickly give us an indication of which breed is the best breed in a specific area. So, thank you.
Yeah, so this is just a little kind of demonstration of all the people who are involved in this project, and then also all the linkages that we've developed from just what was a very small initial project, because my background certainly wasn't in genomics. Kind of visible now, but I've got great people around me, and there's probably a few more dots to put up there on the board now, but yeah, it's kind of exciting times. There's lots of different areas that you can move into, so watch the space, I suppose.